the fluid mosaic model is our basic understanding for how membranes operate and how they're structured. So as I was saying in the previous video, membranes are what compartmentalizes the cell and makes the cell a cell, and what makes the organelles inside the cell sort of separate compartments altogether. So let's take this name apart, fluid mosaic. What does that mean? Well, fluid basically means liquid, that even though this thing forms the structure, the viscosity of it is essentially like a liquid, like olive oil. And mosaic, if you think of a mosaic tile floor or mosaic art, mosaics are made of tiny bits. So we can write that, made of tiny bits or different pieces, right? It's not all one uniform structure. So a couple things about this. Is a complicated image here, but we'll take it apart and do a few important things. So what we're saying then is the top side of this is the extracellular fluid. So we're sort of saying that this is the boundary that makes up the cell. So inside is the cytoplasm then. And that the outer part of this, so the side of the membrane that faces the extracellular fluid, we call it the outer leaflet. Kind of a weird term, leaflet is how we describe that. That includes everything above this, the sort of center. If you were to kind of rip the bilayer apart, you would end up with two different leaflets, the outer leaflet, and then that means that inside, right, is the inner leaflet. And the inner leaflet is what faces the cytoplasm or interacts with the cytoplasm. So a couple things about the way the lipids can move. Like we said, it's the fluid mosaic model. So if you were to pick on, say, a lipid, so I'm going to point at this one right here. I'll color it in. This lipid right here has what we would call free lateral movement. Now, lateral means side to side, and free means that it's capable of doing that. And so it can wiggle, go throughout the entire membrane and wiggle around. And so that's true for all of the lipids and many of the other things in here, is that they're able to move side to side and back and forth then in that plane, staying, for example, within the outer leaflet. Likewise, the lipids and structures that are within the inner leaflet are free to move around as well, because again, they are fluid. But what can't happen, or what requires some extra help, is what we would call transverse diffusion or transverse movement. So that's, for example, if we were to take a lipid and try to get it to go from one leaflet to the other. So if we were to try to convince a lipid that's in the outer leaflet to end up in the inner leaflet, that's going to be difficult. And so what we would say is this has very limited transverse. So transverse means across movement. In other words, what we can say is no flipping. That's what it's called when you go from one leaflet to the other, flipping. However, if you have an enzyme, you can do it. There's a special enzyme that'll do this. So no flipping without that enzyme. And the reason for that is, again, remember, the nonpolar tails are in the interior and the head groups are very polar. So if you're trying to convince a polar head group to migrate through the nonpolar core, that's not going to be favorable and not really going to happen readily. But an enzyme can help that by sort of grabbing onto it and holding it through. So a few other things that we'll talk more about as we just kind of get into other aspects of enzymes and that are involved in metabolism. We talked a little bit about proteins that are embedded in membranes. So notice, for example, here we've got an integral membrane protein or a transmembrane protein that it goes through from, all the way from the cytoplasm to the extracellular fluid here. We've got what we call a peripheral protein, which is kind of just interacting with one leaflet, not really going through. We've got channels that can allow things to go through as well. So we'll kind of explore these a little bit in a little bit here. So that's what I want to focus on as far as the structure. But I want to kind of dive in a little bit deeper in terms of the composition of these lipids and what the membrane can be made of. And so different membranes, different compartments have different roles. The mitochondria, the plasma membrane, the cell membrane, and the endoplasmic reticulum, for example, all have different roles, different things that have to happen. And so the structure and the properties of the lipid bilayer, the membrane, and those features and those structures will change. And what we can vary are things like, for example, the acyl chains, right? So the nonpolar tails, essentially. We could change the length of those. So we could have, 14 carbons versus 18 carbons, right? We could also change the saturation of those. So you could have perhaps a saturated carbon chain, so saturated fatty acid, or you could have, unsaturated. So you can vary that. And that will certainly change some physical properties, right? Longer carbon chains have stronger intermolecular forces, and that will actually change the viscosity of the membrane. And if you have more saturated fatty acids, that's going to, again, have stronger IMFs than the unsaturated ones. Okay? So you can change the acyl chains that are part of the membrane. You could also change the head groups. And so remember we talked about the diacylglycerol phosphate structure. They had different head groups. You could alter the polarity. So you could have things more polar. You could have positive versus negative groups on there. Remember we saw choline, we saw ethanolamine, we saw serine. There's different groups that could be in those on the heads of those phosphate groups, and that can cause very different properties then. So you can change the charge of the membrane as well. So different membranes will have different proportions of these lipids depending on what particular characteristic that it needs in order to operate. And we're not going to get too specific with this, but things that will change would be the fluidity, for example. So like I was saying, if you have longer and more saturated chains, you're going to have decreased fluidity. If you have shorter and more unsaturated chains, that's going to have weaker IMFs, and so the fluidity is going to be even weaker and thinner, less viscous. You could also alter the charge, right? Positive, negative, or just polar. So charge and polarity of the membrane. And a lot of this will end up having effects on the permeability of the membrane. So how readily do things like gases get through, or polar molecules, small molecules, right? If the membrane is more fluid, if there are certain charges, then certain molecules can get through the membrane easier than others. Okay? So that is the our understanding of the fluid mosaic model. And how that is the current understanding of how membranes operate.